flower color. And the flowers that are oftentimes used in this example are called 4 o'clocks. We don't need to know that. In flower color, there are 2 different alleles, and they could be demonstrated as R1 and R2. Oops, these are the R1, and I'll put this one R2 down here. Okay. So R1 codes for red colors, and R2 codes for white. So notice how, since there's incomplete dominance, I didn't put little r and big R, okay? So we could have a true breeding parent for red color, and we could mate it with a true breeding parent for white color. So this would be the parental generation. The F1 generation would be this, okay? And in this case, this is pink. So if Mendel was using 4 o'clocks in these flowers, he might have assumed, oh, well, this supports the blending theory, right? However, one thing that it doesn't support is in the next generation. So if you did another cross, right, of those 2 individuals, you would get red, you would get pink, and you would get white. So in the subsequent F2 generation, right, you would get all that variation back out. So we don't lose the variation, right? Variation is really important. So if you look at this and you did the Punnett square, you could do the Punnett square just like we did the Punnett square with complete dominance. You would actually get 1/4 red, you would get 2/4 pink, and you would get 1/4 white. Okay? So it's actually a 1 to 2 to 1. So it's not a 3 to 1, it's a different ratio, okay? So that is incomplete dominance. Now in humans, there's an example of this in humans, which is called, let's see, topic, whiteboard. Familial, so this is my human example. Familial, whenever you see familial in a word, this means that it is genetic. So it's passed down in a pedigree. And this is hypercholesterolemia. Hypercholesterolemia. And this means that you have lots of cholesterol in your tissues. And this can lead to massive heart attacks, right? And so F1 is normal, right? But the mutation F2 causes abnormal levels, so abnormally high levels of cholesterol. Okay, so most people in the population have this combination, right? However, if there is a mutation that's passed down from subsequent generations, they can have this combination. So it'd be F1, F2, right? And this combination can lead to heart disease in your 20s and 30s. Okay. And then if you're unfortunate enough to have 2 parents that are carriers for this particular mutation, you could actually get 2 copies of the defective allele. And so what that means is that that's a protein that cannot take cholesterol out of the tissues and store it in the liver. And so this actually causes heart disease in children and young adults. So sometimes you hear of these cases where children or young people will like be playing sports and all of a sudden they'll keel over of a massive heart attack. This is that kind of common in this particular phenotype, right? So running around the field and they die, or playing basketball and they die, massive heart attack. It has to do with that particular combination. Okay, so that's an example of incomplete dominance in humans. And so we see how we have more variation than just having the disease or not having the disease. It causes variation in when the disease is occurring. Okay. Are there any questions about that? Okay, So we can also look at, let me go back to my mendelian genetics. We can also look at a phenomenon that is referred to as epistasis. And so these are kind of all these exceptions to our really basic Mendelian pattern of inheritance. And so epistasis is where you have two or more genes causing, or maybe not causing, influencing the trait. So my favorite example of this these days is eye color. And I'm only going to look at brown and blue eye color. So eye color is never as simple as it generally is described, like where you have like just one gene, big B and little b. So it's more complex than that. And so green is actually caused by a different gene that influences how the pigment is deposited in your iris of your eye. And so we're just going to kind of not talk about green eyes at all in this instance, okay? So we have a gene called HERK2. So that, you know, that's the gene, and there are two different alleles. The normal allele is dominant, right? And so I'll just give that an H. And this codes for melanin. Okay. So that is what determines that the brown eye color. So we also have another gene, which is called OCA2. And OCA2, we're just going to say that the normal allele is O, right? And so this codes for an enzyme that is needed for the production of melanin. Right? So these two genes influence whether you have brown eyes or blue eyes. So we have gene interaction, right? The genes are interacting. So we see in most cases, unless you're albino and you have problems with the production of melanin, most individuals are this combination, right? However, when we look at the O gene, they could be this. And so this would produce brown eyes, right? Because we have two dominant alleles, okay? If we look at this combination, however, let's use little o, okay? This produces blue eyes, right? So, I guess I need to put the normal allele produces the enzyme. The little o is the defective enzyme. So this tells me that I have brown eyes, right? And you think that this person would also have brown eyes, but when you look at this other gene, that is the blue eyes, okay? So they can actually estimate when a mutation arises in a population. So they've gone back and they've tried to estimate when this little o mutation arose in the human population, and they estimated it was between 7,000 and 10,000 years ago. So if we look at the headlines you might have seen, okay, so it says blue-eyed ancestors, humans have a single common ancestor. So that means that we believe that that little o, from the big O to the little o, that happened only once, right? And the reason why is this: if we look at the probability of, you know, what are the letters that are changing, you know, they're the same in all people that have blue eyes. And so 6,000 to 10,000 years ago, doesn't seem that long ago. So before that, there were no blue-eyed people, okay, on the planet. And then when you try to look at what the people looked like back then, they tended, where they came from, to have dark skin and dark hair. And so they've tried to figure out what this ancestor had, and this is a drawing, right? So that's pretty startling, right? You can imagine seeing somebody with really dark pigmentation, but then blue eyes, right? So how can, if individuals only had, if this mutation only occurred once, how do we get individuals that have 2 copies of the mutant's allele? If it only occurred once, how do you get people with 2 copies of it? Inbreeding. Inbreeding, excellent. Okay. So actually, we have to have people that are genetically related somewhere along the line had to breed with one another to produce a blue-eyed individual, right? And so if we go back to kind of our look at this, okay. So if the mutation arose only once, so I'm just gonna draw my little pedigree here, and this is an oversimplification, right? But I'm just gonna show you how that would work, right? Let's just say that these brothers and sister produced a child with blue eyes, okay? Probably was more like second cousins and third cousins, right? So this person would have big O, little o, right? This is big O, and then it can pass it on to big O, little o, right? And then we can get an individual with little o, little o. Okay. Right. Now, I'm not saying that blue-eyed people are inbred, right? It's just that human populations, you know, were rather small. Now we have this global, huge population. But if you think about back in the day, you know, it might be that you would have a relationship with somebody that might be your second cousin, or maybe even your first cousin, but somewhere along the line you share a common ancestor, right? Okay, so that's the blue-eyed individual, okay? And that is not all that common, because if you think about it, in the past, there was this idea that inbreeding, and this is called consanguinity. So coming together of sanguine, what does sanguine mean? Blood, right? So consanguinity, so sanguine means blood. So this is coming together of blood. And so this is what we don't refer to as inbreeding in humans, we say it's consanguinity, right? But if you look at Cleopatra's pedigree, right? There's lots of double lines in it, right? And so it used to be that you had royal blood, right? And that royal blood was meant to stay within the family. So you had lots of consanguinity, right? And so we might look at this and think, oh, this is really bad, right? But we actually do this in our purebred animals, right? So if you think about animals like dog breeds or horses, where we're breeding individuals that are similar, right? We're actually producing organisms that are more likely to have recessive traits expressed. So like in purebred golden retrievers, they have a mutation that causes them to go blind, right? They get juvenile cataracts, like cataracts as puppies. And that is a recessive trait that showed itself because we have been inbreeding those animals to produce the ideal, you know, that dog that has those characteristics, right? So mutts tend to be much healthier than purebred dogs because purebred dogs have recessive traits being shown. Okay, are there any questions about that idea? Okay. Plyotrophy. This is a big word, but what it means is that one gene can have many effects on the phenotype. So it's actually kind of the opposite of epistasis. One gene has many effects. And a good example of this in humans is a disease called Marfan's syndrome, where individuals produce defective collagen. And what type of organic molecule is collagen? Should know this for your midterm. Is it a carbohydrate, protein, nucleic acid, or lipid? Does anybody know? Nope. Nope. Protein. Okay. So it is a structural protein, right? So collagen is like everywhere. It's in your bones, it's in your blood vessels, it's like everywhere. So if you produce defective collagen, you're going to have multiple symptoms, right? It's going to be like everywhere you have collagen, it's going to have an effect. So these individuals tend to have these characteristics, right? So they tend to have eye problems. They tend to be very, very tall, and they have heart and lung problems, and they have very long arms, fingers, and toes. So what do you think? There was a president that we think has this particular, or had this particular, is dead now. What president do you think? Abraham Lincoln, right? So they think Abraham Lincoln had Marfan's syndrome. And he might have not lived too much longer than he already did, because he probably was going to have massive heart attack because of defects in his heart. Okay? So that's the idea of plyotrophy. Okay. Okay. We also have what is called polygenic inheritance. And this means that we have a trait caused by many genes. So many, so not just 2 or 3 or 4, but many genes influence a trait. And one example of this is skin color in humans. And so it's believed that there are 6 different genes that code for pigments. And so I'm only going to talk about 3, and I'm just going to show you how it works, okay? So let's just simplify it to 3 genes, where A, B, and C produce melanin. A, B, C produce melanin. So I'll just put 3 gene model. Okay. Little a, little b, little c produce no melanin. Okay. So what this means is that you can have an individual that is very, very dark, like this, has this combination, right? And an individual that is very, very light with this combination. And they would have an offspring that is intermediate in color. And that makes sense to us, right? We're like, oh yeah, I can see that. I would predict if we had a very dark father and a very light mother, you'd produce an offspring with some more intermediate, right? But notice how if you have people that are intermediate, or even, you know, they might even have more dominant or more recessive traits, the way that it is genetically recombined is we can get lots of variation, right? So we could get, potentially, if we crossed 2 individuals that were of intermediate color, we could get potentially an offspring that was pretty dark or very dark, right? We could also, if this one inherited a little a, little b, little c, little a, little b, little c, right? Very, very light, right? So what this means is that 2 people of intermediate color could produce very light offspring. Okay? And that, in some cultures, is kind of confusing, right? So if we look at the range, it tends to have a normal distribution. So the possibilities tend to be somewhere here, where you'd have very dark offspring, and here you'd have very light. But most of the time, you have offspring that are intermediate in color. Okay. So if we look at this in your book, it looks something like this, right? So they talk about the P generation, they talk about the F1 generation, those are the hybrids, and then we can see that we can get lots of variation in color, okay? So Interestingly, sometimes we get this happen, right? So these are the parents, and these offspring are both their children, right? So these are what are referred to as dizygotic twins. So they're fraternal twins. So they're related like they would be brother and sister, right? They're twins, but they are much different in color, right? And so that is expected by the way that Mendelian inheritance occurs. Okay. So that's polygenic. And you might have heard a few years ago, this got a lot of concern in European cultures. And so in Europe, they have a group of people that are called the Roma, which are kind of negatively termed the gypsies, right? So the Roma are darker skinned than the white Europeans. And there were some countries that you had people going into the Roma population and stealing the white-skinned offspring because they believed that they must have been stolen from, they didn't believe, so for example, with this child, they didn't believe that these were her relatives, right? So they took them back and they said, these can't be your children because of them, right? And so there's this huge, huge big deal about it, right? And so that is an example of what the variation you can get in polygenic inheritance and how it influences the culture. Okay. So with polygenic inheritance, we don't have discrete variation, but it produces what we would refer to as continuous variation. Versus discrete. Okay. Okay. So this was another example of the same thing. In apartheid Africa, oftentimes you'd have white people and they would give birth to a child that was darker skins, and they couldn't put on their birth certificate that this is their child. They couldn't put on their birth certificate that this child was white. They had to say that that child was black. And so this child didn't have the same advantages as if she were white skins, because she couldn't go to certain schools and she couldn't, go to certain places because of the way the government ruled and discriminated against that population. Okay? So you can have that problem too. This is actually from a movie called Skin, which talks about two white people in Africa, South Africa, that have a black daughter. Okay. So we have been talking about traits that are on non-sex chromosomes. So now we're going to look at sex-linked traits. And so these are traits that are determined by genes on the X or the Y chromosome. Okay, so determined by genes on the X or Y chromosome. Now, the Y chromosome is tiny compared to the X chromosome, and it seems to be getting smaller with evolution. Some people hypothesize it's actually going to go completely away. But the X chromosome is much bigger. It has many more genes, right? So generally, when we're talking about sex-linked, we're talking about X-linked. So X-linked because the X chromosome has many more genes. So can anybody give me an example of a trait, a disease, we'll talk about, a disease in humans that is sex-linked? Does anybody know, like, or maybe a trait? Color blindness. Color blindness, excellent. Okay, so let's look at color blindness. And this is specifically red-green color blindness. There's different forms of it. Okay. And so in this particular instance, we have to say that the gene or the allele is on an X chromosome. So this is X with a big C, and that is normal. Okay. And X with a little c and I'm put that my line over my little c so I can tell, this causes the trait. So this would be causing the color blindness. Okay. Now, because it's on the X chromosome, this means that females can be carriers, but males can't, right? So females can carry the trait, males can't. So it's actually, probability-wise, it's more common for a male to be colorblind than a female, but females can be colorblind, okay? So let's look at how this would play out in a pedigree. So we have generation 1 and 2, right? And then we could have a child with color blindness. Okay. And then we could have a child without it. And then they could have, let's say, a child with, say a girl with. So this one's got to be it too. So I'm just going to show you how it could theoretically also be found in females. Is there anybody in here that has red-green color blindness? No? Okay. Or you don't want to admit it. Okay. Okay, so the first thing we have to do is we have to tell myself, just remind myself, which one are males? Are they squares or are they circles? Squares, right? So I would use a Y. I'm going to use a different color. So I would put a Y next to all the males, just so that I remember if I'm doing this problem on my own, I'll remember those Y's go to next to all the males. Okay. Then what I'm going to do is I'm going to say which males have color blindness, and meaning that they have to have a little c. And that would be these two. So I don't know if you can see, but my C's have little lines over them, okay? The male that doesn't have color blindness would have to have a big C. Okay, that's a big C. Okay, so this female has a son with color blindness. So what combination of chromosomes does she have to have? She's a carrier, right? She's got a big C and she has a little c. Okay, and I know that by working backwards, right? I say, oh, well, she had a son that had it, so she has to be a carrier. Okay, same thing with this one. This individual has to be a carrier, right? And so I can go ahead and mark that in. She doesn't have it, so she has to be a carrier, right? And so then what is the female that has color blindness? Right? So this just shows that you can get a female with color blindness, but it's less likely because you have to have a father that has color. She has to have a father that has color blindness. And she has to have a mother that is a carrier, right? So it's actually the coming together of two families that have this mutation. Because this guy, this individual is from another family that he marries in. Okay. Okay. So the question is, if they have another child, what is the probability that that child will have color blindness? So the way to set this up is to figure out what the genotypes of the parents are. So I would write my X and my big C, my X and my little c, right? And then the sperm would have to be X, little c, and Y. Does everybody see where I got that? So this is just the parents, and these are the gametes that they could produce, okay? So if they have a female child, X, and I always put the dominant one first, okay? So if they have a female child, then it is 50% chance that that offspring would have color blindness. And then if I look at the males, in this case, it's the same probability. I always put the X's first too. So in this case, it doesn't matter whether they're male or female. They would also have a 50% probability. Okay? So you have to kind of remember that when I say it's X-linked, you have to remember that, oh, now I have to pay attention to the sex of the individuals in the pedigree. Then I kind of have to pay attention to what the X chromosome, the variation of the X chromosome, and I put in my Ys, and then I can kind of add to it through processes of deduction. Any questions about that idea? Let's see what else I have here. Okay, last thing. Xinactivation is a really interesting phenomenon that occurs in males. So, oh, excuse me, only occurs in females. Sorry, it occurs only in females. Right? And the reason for this is that females have 2 X's, right? So that means that they have twice the amount of genetic material than a male, because males only have 1 X. So what has to happen is one of the X is turned off, right? So we turn off 1 X chromosome. And this will turn off 1 X chromosome. And this X chromosome becomes what we call a bar body. And bar with 2 R's, it's called a bar body because it's a darkly staining structure in the female's body cells, okay? So when we look at X inactivation, one example of this is calico coloration in cats. So if you find a calico cat, what sex is it? Females, right? So generally, unless there's some weird mutation, all calico cats are going to be female, not male. And the calico that I'm talking about is the orange brown or the orange black. The white comes from another gene that interacts to cause white. So we're talking about like orange black. So it looks more like this, right? So this is my calico cat, okay? And so they have an X with a big B, right? And that codes for brown fur. Brown fur. And then they can have an X with a little b that codes for orange fur. Right? So as these embryos, cat embryos, develop, there's a random probability that the little b will be inactivated or the big B. So it's just kind of like random inactivation. So if we look at what happens is we start out with both of them active, right? In one group of cells, the brown one stays active and the other one forms a bar body. So this would be brown fur, right? In this one stays active and then the other one is the bar body and this would produce orange fur. This process is random. So generally you'll never find 2 identical calico cats, right? Because this is actually during development. It actually doesn't have to do with genetics. It's actually random X inactivation as the embryo is developing, early on during embryonic development. So all of these cells that come from this are always going to have this one active and this one inactive, and same thing with this. So all the cells from then on out have that inactivation. Okay. Now there is an example of this in humans, but rather than fur color or skin color, it has to do with sweat gland distribution. So in humans, there's an example. So some people have a mutation that determines that they're not going to produce normal sweat glands, and so they have this pattern due to X inactivation where some of their skin won't sweat and some of their skin will sweat, right? And so I actually knew somebody like this when I was a kid. She had like a line down her middle of her body, and this side would sweat, and so like her t-shirt would be all soaking wet after, doing sports, and this side wouldn't sweat, right? And so that had to do with X inactivation. Okay, So those, we'll put the bar bodies here. So these are bar bodies. Right, those are bar bodies. Okay. The bar body is just the X chromosome all packaged up. Okay, so let's look at your review sheets. Hopefully maybe you brought it, or you can just write what I'm going to say. I just want to make sure that I went through everything that's on it. And I didn't, so that's good. Okay, so looking at the back of the sheets, right? We're going down. We didn't talk about inbreeding depression really, so you can cross that off. But I did mention that purebred dogs tend to have more illnesses than others, and that's an example of inbreeding depression. We just talked about Xinactivation, Plyotrophy, right? That was Marfan syndrome. We did not talk about linked genes, so you can cross off linked genes. We talked about polygenic traits like skin color. We did not talk about epigenetics, so you can cross that one off. We'll talk about that after the test. And we did not talk about the genetics of cancer, so you can cross that one off as well. And then remember on the front, remember to cross off the nitrogen cycle. So in that first group, near down the bottom by ocean acidification, you do not need to know the nitrogen cycle. So please bring a pencil. I'm going to provide Scantrons. You don't need to bring a Scantron sheet, but you'll want to bring a pencil so that you can fill out the Scantrons. And so I will, also, I wanted to let you guys know that rather than having lab next week, I'm going to hand out homework. And it's going to be genetics homework. And then we'll just have open lab. So if you want to come and do the genetics problems with me in lab, I will be there. Otherwise, you can just do them on your own. And then you're going to put them in your lab notebook, and I'm going to grade them just like a lab. So next week, you can, if you like, need to be someplace at 2 o'clock. Like Halloween party, right? 2 o'clock, starting at 2 o'clock. We're going to have a homework assignment.